So hello once again, how are you? This is Mr. Gos Biolectures taking you to Biolectures. These biolectures are very important, so make sure you subscribe, like and share so that you can see more of these videos. So today's biolecture, you're going to look at something called the Krebs cycle. This is also called like a carboxylic cycle. It takes place in what we call the mitochondrial matrix. We saw that of course in the other video. You can go and watch it if you not watch it. But as now, without wasting time, you're going to see what we call the Krebs cycle. We want to see how energy is yielded through this cycle here called the Krebs cycle. Now, I want us to look at something interesting here. The whole of this thing we are seeing here, the whole of this one, is what we call the cell. The whole of this one here, what we call the what? The cell. It is a human cell. Now, or a plant cell, but measure this is what we call human cell. This is what we call human beings. Then we are seeing something inside here that is very interesting. This stuff interesting here that is inside here. This one here with some structure here. This is what we call the nucleus. What we call the nucleus of the cell. By having what we call the DNA, and this is what we call nucleus. It's what we call the nucleoplasm. Now we have what we call this liquid inside here, which what we call cytoplasm. This is called what? Cytoplasm. I want you to watch clearly because I don't want you to get lost. Then within the cytoplasm, we have what to call this structure we are seeing enclosing here. The whole of this one here enclosing here. That's what we call the mitochondrion. It is called the mitochondria, the powerhouse of the cell. It is called the mitochondrion. This one here. I'm going to just label it somewhere. Let me put it somewhere here. It's going to be called what? The mitochondria. I'm going to abbreviate it. The mitochondria. This one here. The one we are seeing this structure here. Now, I want us to begin seeing how is pyruvate, because we saw in glycolysis that during glycolysis, the process ends with a 3-carbon compound, which is called what? Pyruvate. Now, this pyruvate here, remember glycolysis takes place in the what? In cytoplasm here. There was glycolysis here, which took place cytoplasm here, giving what we call a 3-carbon compound called what? The pyruvate. That's something very interesting. So this pyruvate here has to move from the cytoplasm and it goes into what we call the mitochondrial matrix here. But we have to first see how does the pyruvate move across the mitochondria to get into what we call the matrix. Let's see here. I've extracted this structure here, this one here. Hope you are seeing this. I've extracted that region there. Hope you are seeing that region that I've extracted there. I want to look at it, examine it clearly. Now, this region you are seeing here, it is the one I've extracted, this one here. Hope you are seeing it. So when you bring it here, it is this. The real structure is this one here. It's the same as this. This one here. This one here. So this one, what called the cytoplasm. The same as this was also what called what? Cytoplasm. Then you have this structure here, this one here, this one here. This one is what we call the mitochondrial membrane. But remember we said that mitochondrial membrane is having 2 layers. This is mitochondrial membrane. It is having what we call this inner layer. It's what we call the outer layer. Then what we call the mitochondrial what? Space. Now this one and this one plus this one in the middle, all of them are categorized in what we call this. So this one is the same as this structure here, which is what we call the outer mitochondrial membrane. Something very interesting. It's called outer mitochondrial membrane. Then you have what we call this, what we call intermembranous space. Intermembranous space. Then it's what we call now the inner mitochondrial membrane. Something very interesting. Then it's what we call the matrix. This one is the same as this structure here, this one here. Same as the matrix here. This is called the matrix. Hope you're seeing that. This is called what? The matrix. So, our major factors want to see how does the pyruvate move from the cytoplasm into what we call the what? The matrix here. Let's see here. Now, this is cytoplasm here. We have what we call a lot of pyruvate here. The pool pyruvate is here. And I'm going to donate it using what we call certain molecule here. Let's call the pyruvate here molecule, which is a what? A 3-carbon compound. Pyruvate here. Now this pyruvate here, what is it going to happen here? It is going to bind onto what we call the receptors here. What's the name of these receptors here? Now on the outer membrane of the mitochondria, we have what we call certain gates. Because this pyruvate, these are tiny molecules. They are like in form of ions. And then these ions here are transported by what we call the voltage dependent anion channels or carriers. These are called what? These are called the voltage dependent. The dependent anion carriers. Also abbreviated called the VDAC, that is VD, like that, the VDAC. These are for transporting what we call the pyruvate and also some ATP and also the ADP. It moves through what we call this, the VDAC, the voltage dependent anion carriers. Now what's happening is that this pyruvate is going to bind here. After binding here, these gates are going to open. When they open, it is going to move. It is going to move. It is moving across into what we call intermembranous space. Reaching the space here, now another problem has come. It has to move across this what called inner mitochondrial membrane. But on the inner mitochondrial membrane here, it is almost impermeable. It is almost impermeable. Just only tiny molecules can pass through. What's going to happen here? Now the pyruvate which has passed through this one, which is here, after binding here, remember we have what called the hydrogen ions here, and also what called the pyruvate here. Pyruvate, these ones are what? These ones are negatively charged. So what is going to happen is that these two here are going to bind into what we call what? The receptors. And then they're going to be transported together. A process what we call electrochemical process. In other words, they're going to follow what we call the symport or the co-transporter, whereby two different ions are moved together in the same direction. So they're going to move by electrochemical process, and then they move towards this, like this, and then reaching here. They're going to reach here. I forgot to call the pyruvate. So to call the what? Pyruvate. The pyruvate is now what to call the what? In the matrix. So here we see that the pyruvate has moved from the cytoplasm into what to call the what? The matrix. But we also have to note that what's the name of these channels here? These channels here are called the mitochondrial pyruvate carriers or channel complex. These ones here, I want to put it somewhere here. These ones are called the pyruvate. Pyruvate. It's supposed to be the mitochondrial pyruvate. Mitochondrial pyruvate carriers complex. So normally abbreviate them as something like the MPC complex, like this one here. So this complex here, when we say complex, that means it's made up of some subunits, some components. So the component that we have here, we majorly have what we call the two subunits. We have what we call the first subunit here is what we call the MPC1. Then the second subunit, what we call what? MPC2. This is what we call a mitochondrial pyruvate carrier, plus another mitochondrial pyruvate carrier to form what we call the mitochondrial pyruvate carrier complex. It is this channel here which is allowing the hydrogen ions and the pyruvate to move along their constituent gradient into what we call the matrix. Because in the matrix here, we have less pyruvate. Why? Because most of it is dehydrogenated into what we call the citrate by the dehydrogenase enzyme, the pyruvate dehydrogenase enzyme. That means you're going to not find a lot of what? Pyruvate there. So I've seen that now. We've seen that pyruvate has moved through the VDAC and also through the MPC transporters to go into what we call what? The matrix. Now, we have seen now what we call pyruvate has gone into what cytoplasm now. Sorry, the matrix. It is in the matrix now. The next part is this pyruvate here is supposed to be converted into what we call a 2-carbonic compound, because this pyruvate is what we call a 3-carbon compound. So we have to decarboxylate it. This is what is going to happen here. I want to show you how we are going to decarboxylate this one here. So we can form what we call acetyl coenzyme A. Now, acetyl coenzyme what? A. So what are we going to do here? We're just going to, because this one here, the one we are seeing here, is the same as what we call the outer mitochondrial membrane. This one here, what we call the outer mitochondrial membrane. Sorry, the inner, sorry. It's supposed to be the inner mitochondrial membrane. The same as this one here. Is the same as this. Then this one, what we call the matrix. This is what we call the what? The matrix. Something interesting. This is called what? The matrix here. What's going to happen here, remember? Something very interesting. Now, this pyruvate here, the pyruvate, is going to enter into what we call the matrix here. So what we call the pyruvate. The 3 carbonic compound is going to enter into what we call the what? The matrix. On reaching here, we have what we call several enzymes. These are called the pyruvate dehydrogenase complex. They are around the 3 enzymes. They are called pyruvate dehydrogenase what? Complex. We have what we call the first one here. We have the second, and also what we call the what? The third. This first one here is having something very interesting. It is going to react with this pyruvate here and then convert it into something that we're going to call, what is this one here? This one is something you're going to call what we call the hydroxyethyl. Hydroxyethyl, something like hydroxyethyl, something like this. Hydroxyethyl. TPP. So this is the first enzyme which is going to convert this pyruvate in what we call the what? Hydroxyethyl. Now this enzyme is what we call the pyruvate dehydrogenase. So what we call the pyruvate what? Pyruvate dehydrogenase. I'm not going to talk about this, the actual the products here, but what we are doing here, we are trying to convert the pyruvate into what we call what? Acetyl coenzyme A. But as we're doing this one Z, what is going to happen is that before we form the acetyl coenzyme A, there are some intermediate compound that we normally form before the acetyl coenzyme A. Now, this one called pyruvate dehydrogenase. Now, the second enzyme which is called E2, it is going to convert now this hydroxyethyl TPP is going to remove this TPP from it here and then forms something very interesting. What's the name of this compound here? It is called what we call acetyl thioester of the reduced lipoic acid. Now, that means we are forming, let me see another compound here. This one is going to be converted into something here. Now, this is the second product we're going to be formed here. Using which enzyme? This enzyme here is what we call the trans. So what we call the dehydrolipol. Dehydrolipol transacetylase enzyme is going to convert this one into another form, another intermediate. Now, then this second product here is going to now be converted into the final product, which is called acetyl CoA. Which enzyme is this? The third enzyme here. The second enzyme was this one here. We have seen it here. The second enzyme. Then the third enzyme is going to convert this second one into the third one, which is the coenzyme A. What's the name of this enzyme here? It's what we call dihydrolipol. Dihydrolipol dehydrogenase enzyme. This enzyme which is going to convert now the remaining intermediate to what was the coenzyme A. But now you don't have to cram these ones here. What you have to know is that the pyruvate is going to be converted into what we call acetyl what? Coenzyme A. I was just showing you how something takes place, so that you should not cram. Now let's go to the real process that are taking place here. Well, don't want to waste time. Of course, we are going to begin from what we call the pyruvate. The pyruvate has moved into what we call the pyruvate has moved into what we call from the cytoplasm into what we call the what? Into what we call the matrix. This is the pyruvate here. This is the matrix. What's going to happen here? This pyruvate here is going to be converted into what we call is going to be converted into something very interesting here, into acetyl coenzyme A. Now this coenzyme A, what's going to happen here is that to convert this pyruvate here, there must be raw materials where we have what called the this oxidized NAD is going to react what we call pyruvate. So when you combine this NAD plus pyruvate, and then it is going to remove the hydride ions, and then you combine this with it to form what we call what? The NAD H2 or the NAD H. This is the same as reduced what? Reduced NAD. This is what we call oxidized what? Oxidized NAD. So then it is this pyruvate is also going to lose a carbon. That means it's going to what we call a carbon by using what we call carbon dioxide gas, which is called the carbon what? The carboxylation. Forming what we call acetyl coenzyme what? A. Acetyl coenzyme A. This acetyl coenzyme A, what's it going to do now? Remember, we have what we call the oxaloacetate. Oxaloacetate. A. This given organic compound here, it is a 4-carbonic compound. What happens is that this 4-carbonic compound here, it is going to combine what we call the what? Is going to combine, this one is going to combine with this acetyl coenzyme A to form a given product here, which is called a citrate. Which is called what? Citrate. Now this citrate here, which enzyme? There must be an enzyme in this mechanism here to bind this to this 2-carbonic compound, to bind it to what we call the what? The 4-carbonic compound. What's the name of this enzyme here? This enzyme is what we call the citrate synthase. It is called citrate synthase enzyme. It is called citrate synthase enzyme. Synthase enzyme. Something very interesting. This is the enzyme here that we are seeing here. Now, so beautiful. Now, what's going to happen is that this enzyme here has what we call the activity of the enzyme to combine the 4-carbon with the 2-carbon form what we call the 6-carbon compound has certain inhibitors and also what we call what? Activators. Now, what can cause this enzyme? What can cause the conversion of this oxalo and this acetyl coenzyme A? What can cause not to form what we call a citrate here? We have what we call inhibitors. One is what we call the ATP. That means increased ATP levels in the blood or in the body. That's one. Second, we have what we call the succinyl coenzyme A. That's increased succinyl coenzyme A. And also we have what we call increased one. These are the major ones. I think these are the major ones. And also maybe some bit of the calcium ions. But these are the major ones here. Now that means that what happens is that if the ATP is much in the blood, it goes on certain receptors on the what? On this enzyme here. It goes and binds, sends signals to the receptors, and tells these receptors here the enzyme to change what we call the conformational structure changes. That means that when the active sites change, that means rate of the catalytic combination of this with this is going to be altered. Hence, it's not format called a citrate. But that's what we have to call what? Inhibitors. Also, inhibitors. What about the activators? We have what to call the low ADP. Low ADP. If you have what to call low ADP in the blood, that means that there's going to be, of course, the succinyl coenzyme A also binds here. It behaves in the same way like ATP, like this. Then we have what to call now the ADP, that's decreased level of ADP. This one is also going to bind on what to call what? On receptors here. And then it's going to cause the increased enzyme reaction format called the citrate. That is it now. Let's go to something interesting. We want now to convert the citrate into another compound. What's the name of this compound here? This compound is what to call the isocitrate. What to call isocitrate. What happens, members here, there's what to call isomerism that takes place. The carbons are the same, they are still 6 carbon atoms. What happens is that what went wrong here was what to call isomerism took place here. Isomerism. The arrangement of the number of the hydrogen and oxygen atoms. Now what's going to happen here? Which enzyme has catalyzed what to call this process, what to call isomerism here? It's what to call isocytase. This enzyme what to call what? Isocytase enzyme. This enzyme here catalyzes the conversion of what to call a citrate into what to call isocitrate. Something very interesting. What is going to happen here? What can inhibit the activity of this enzyme isocytase to convert citrate into what to call what? Isocitrate here. Something very interesting. Now, we have what to call certain poisonous molecules, like what to call the fluorocitrate. The fluoroacetate compounds. Now, what happens is that any compound that is informed what to call the fluoroacetate, it can inhibit the activity of what to call what? The enzyme. What happens? For example, it goes and binds on what to call the receptors of this enzyme here, and then the conformational structure is changed, and therefore this enzyme can no longer activate the conversion of citrate to what to call iso. That means if you cannot form this, it means you not get the energy. Even what is not going to take place, meaning no energy. Now this explains the reason why. Did you know why the poison normally kills you? Now we have what to call some poison. If you accidentally swallow poison, what is going to happen here? Most of these poisons here, they are fluoroacetates. Now when you swallow it, when it goes into the blood, this fluoroacetate here, the blood or the body immediately converts into what to call the fluorocitrate. This fluorocitrate is very, very poisonous. Very, very poisonous. You get? It immediately goes and binds on the receptors of this enzyme isocytase, and then it stops further ATP manufacturing or synthesis, and therefore you will not have energy. That means also the heart is not going to be pumping because it's not receiving, it's not getting energy. The muscles are going to not to contract because it hinders the flow of the calcium ions. Listen here, what can you do in a case by mistake you happen to swallow that or that poison? This is what you do. Just go in any shop and get what we call the sodium citrate, succinate. What we call now, how to overcome it. That's what we call the solution now. Just go and get what we call sodium succinate. And also what we call the calcium gluconate. This sodium succinate here, what does it do? It is going to bypass. Instead of the reaction converting from this to this here, this one is going to take the low sodium citrate of the citrate here, and then it converts citrate here directly to what we call the fumaric acid. We say it has bypassed. Just like if you can be having someone, maybe this person here, someone is maybe you're playing a football match or in a game, and someone says, I'm not going to play the game. The person refuses, then you say, no, we can't leave the game not to be played. The another person comes in says, I'm going to take over that one's role, or maybe the goalkeeping, we say you bypassed what someone was going to do, so as the game can go on. So this is what called sodium succinate. This one is going to bypass. Then what called gluconate. This one is just going to maintain the calcium levels in the blood, so as you can have enough calcium, so as you can have what you call body functioning. Muscles are going to be contracting, getting energy, heart is going to be pumping, and then also liver is going to be functioning. The brains are going to be functioning. You get? This is now the medicine you can get. That's if the poison is not that much. But if the poison is extremely too much, we can give you this one here. But again, we do what to call the hemodialysis. That's one. Also, we can do what to call the filtration. Also, we can do what to call the perfusion, something like this. So you can do any of these ones here in support with this. That is in case if the poison is extremely high. Now, we are going back from this format because isocitrate here, 6 carbon compound. What's going to happen here? We are going to now convert this isocitrate into what we call what? A given compound called the alpha ketoglutarate. What's going to happen here? Now, this process is going to undergo around like 2 processes here. We are going to lose a hydrogen, which is going to be now dehydrogenation, whereby the NAD, oxidized NAD, is going to receive the hydrogen ions, and then it's going to be converted into what we call what? The reduced what? NAD. At the same time, this one is going to lose a carbon. The process called what? Use the carbon, also the process called the carboxylation. But which enzyme is going to be here? It's what called isocitrate, dehydrogenase enzyme. Isocitrate, dehydrogenase. Dehydrogenase what? Enzyme. Something very interesting. Let's continue. Now, I want us to look at this. What are the inhibitors for the conversion of isocitrate to what to call what? Alpha ketoglutarate. What can inhibit this enzyme here? One, we have what to call, there are some receptors here. We have what to call the increased levels, what to call the NAD, H. If you have a lot of, that means that you're going to have a lot of energy. That means this is going to send signals that are going to bind on what to call what? These receptors, or the enzyme of the receptors, causing it to stop the conversion of this into this. Second, you have what to call the calcium levels. This is an inhibitor. This one is inhibitor. You have what to call now the activators. Activator, how about to call now the calcium levels. Calcium, that is in case you don't have enough ATP, what's going to happen is that signals are going to be sent on what to call the given receptors, and then calcium is going to bind here, causing what to call contraction of the muscles, so that you can get more energy. Then also we can have another inhibitor. We have what to call, if you have what to call increased what? ATP. Then another, this one is going to be inhibitor. Then another activator is going to be what? Low levels of what to call what? ADP. Something like that. Now, that is the reaction we're going to form here. Now, this compound is how many? Because you lost a carbon, they are now going to remain how many? Going to remain 5. This is 1, 2, 3, 4, 5. This is 1, 2, 3, 4, 5 carbon atoms. There is something interesting here. What's going to happen here on this alpha ketoglutarate? This one here is going to lose a carbon atom. That means it's going to undergo what to call decarboxylation, losing a carbon dioxide gas. At the same time, it's also going to undergo what to call some form what to call dehydrogenation, losing what to call the hydride ions. Received by what to call the NAD plus, form what to call the what? The reduced what? NAD. Now what's going to happen is that the reaction here to form something here was in this format here. We shall have what to call the alpha ketoglutarate, keto, then plus what to call the coenzyme A, then plus what to call the NAD. This is going to give us what to call now the succinyl. succinyl coenzyme A. Then this one is going to, this one has bond here. Okay, has formed something else. Then it has a format to go plus what? NAD. They reduce what? NAD. That means now we're going to form something interesting here. This is going to be the succinyl coenzyme A, you get, which is now we have lost one carbon, which is supposed to be how many? This is going to be 4. 1, 2, 3, 4. The 4 carbon what? Compound. Something very interesting. Now, what can also affect this one here? We have what called inhibitors. Inhibitors are almost the same. We have what called the NAD. NAD is when there is, this one can inhibit in case there is high levels of this. High levels of what? This can affect the function of this enzyme here. But what's the name of this enzyme? I don't talk about this here. I mean, this one enzyme here. Let's talk about it. High levels of this can cause the inhibition of that enzyme here. What's the name of this enzyme here? This enzyme is something very interesting here. The enzyme is what we call the alpha keto, what we call the alpha keto glutarate dehydrogenase. Something very interesting. You can also what to call the reductase dehydrogenase enzyme. Now that's what we call now the enzyme involved here. Now there's something interesting here for enzyme A, something like this. Now so I call enzyme A, something like this. This just having what we call the phosphate here, and then this coenzyme succinyl supposed to be a succinyl coenzyme A. It is having what we call, remember it was having how many carbons? This one was having 6, 5. It's having what we call 4 carbon atoms. What is going to happen that this succinyl coenzyme A is going to lose what we call 1, the coenzyme A. That's the first thing. Secondly, it is also going to lose what we call a phosphate. It's going to be transferred, and this phosphate is going to be accepted or receive what we call the ADP to form what we call what? Form what we call ATP. The first energy ATP molecule that is yielded directly. Apart from this, this ATP can also be received by what we call the guanosine diphosphate. And then this guanosine diphosphate is going to be converted to what we call what? Guanosine triphosphate. This is also an energy store or energy carrier. That means that this one stores energy just like also the ATP. Except this GTP here does other functions apart from storing energy, like the protein synthesis and so on, also acting as binding sites and very many others. So what's going to happen here? We lost the CoA, we have gained an ATP, then which enzyme is going to participate here? This enzyme, what we call the succinyl, this enzyme you are seeing here, called the succinyl synthase. Citrate enzyme. Now from what we call the succinate. Succinate, this is a 4 carbon compound. What's going to happen with succinate here? This succinate here just going to undergo what we call certain reactions. For example, like the dehydrogenation is going to take place here, whereby it's going to lose certain kind of the hydrogen ions, which are going to be actually what we call the NAD plus, from what we call the what? The reduced NAD, H2. That's very interesting. Then, so from what we call the fumarate or fumaric acid. This fumarate here is just going to undergo what we call certain reactions, whereby we have this enzyme here. What's the name of this enzyme here? The same enzyme here is what we call the succinate dehydrogenase enzyme. The succinate dehydrogenase enzyme. From what we call the fumarate. The fumarate is just going to undergo what we call certain form of what we call dehydrogenation. That's from what we call. No, this is just going to add some water into this reaction, and then we shall see that fumarate is going to be converted into what we call the what? The malate. Using which enzyme? This is also a 4 carbon compound enzyme. This is what we call the what? The fumarase enzyme. there is something important you have to note. How can you know that someone is having what we call tumors in the body or in any cells? Now we have this fumarate here, an enzyme converting fumarate into what we call what? The marate. Now if someone is having a lot of fumarate and it is not converted, that means this person here could be having damaged cells. Another one is tumors. We have what to call types of tumor. We have what to call the benign tumors. We have what to call the malignant tumors. Now these malignant tumors here are the ones normally cause what to call the cancer. They're same as cancerous tumors. Then we have what this one is here and non-cancerous tumors, because for them they just cause what to call the improper growth of the cells. This one is here. They don't affect what to call the hormones. It means that if you have a problem with this one, benign tumors, it means that you just have an imbalanced hormones. For example, you have your body secreting a lot of norepinephrine and also norepinephrine, and then you see the heart rate is going to not balance. The heart can beat a lot or it can beat slowly. You see what called unexpected circumstances are going to come in if you have these tumors here. So if you have what called high levels of what called the fumarate, it is indicating that the cells got damaged or they are now swelling from what called the tumors, and therefore the fumarate cannot be what? Broken down. You must know that. Then we go to this fumarate is going to pick one of the marate. You have seen that. Then the marate is supposed to be converted into what to call oxaloacetate, which enzyme this one here, what to call marate dehydrogenase enzyme, the marate dehydrogenase enzyme. At the same time, for this enzyme here, we shall see that there's going to be some loss of converting this into the malate. We shall see that converting the malate into what we call the OAA. We shall see that we are going to lose some hydrogen ions. It's supposed to be what we call the hydrogenation, losing some hydrogen ions like this, from what we call the NAD. It's supposed to be what we call the what? The NAD, oxidized NAD, from what we call the NAD what? H. So this is just a recap of what we call the Krebs cycle. Just a brief summary of this, a brief summary. We have seen that the Krebs cycle, takes place in what we call the what? The matrix. Then the matrix here, what was taking place? There was what we call the substrate, was what we call the what? Substrate was the pyruvate. Then what's the end product that we formed? Was what we call the what? This is a 3-carbon compound. We have to call now what we call a 4-carbon compound, which is called what? Oxaloacetate. How many ATP did we yield? Supposed to be 1, but remember these are 2 because we have 2 pyruvate. These are the 2 molecules of what we call ATP. What about now NAD? We have what we call 2 molecules of what we call what? NAD. There is this one here. This is 1. Also we have this one here. This is 2. And also FAD. That means FAD. Also what we call 2 molecules of what we call the what? The FAD H2. And then you have seen the enzymes taking place there. You don't have to just cram this, but you just have to understand. You get the point. Otherwise, thank you very much for watching this. I think you must have acquired something. If you have obtained something from there, just subscribe, like, and share. You can also ask in the comment section. You get something very interesting. There is something I want to talk about before I go. We want to see what is the importance of what we call the citrate, and we have what we call the clinical relevance. Clinical relevance. Now listen, members. How can you prevent or how can you control something normally called what we call the renal tubular acidosis? Renal tubular acidosis. This one is a disease of what to call the kidney. Which medicine can you use? We have the medicine here. What is that? It is called the citrate. This citrate here can treat what to call the kidney renal tubular acidosis. For example, what to call the potassium citrate. Potassium citrate. So if someone is having the renal tubular acidosis, in other words, something like stone in the kidney stones, or within what to call the kidney tubules, you just have to take what to call the potassium citrate. It is going to neutralize the so-called what to call the kidney alkalina. It is called the what? The kidney alkalina. Alkalina. It goes and neutralizes what to call the acid of what to call the what? The tubules, and therefore someone becomes normal. And where do we get these citrates here? They're just from what to call the fruits, citrus fruits. How about to call, for example, how to call the oranges, the lemons, how about to call the limes, and so on. But also some fruits that are having water, like the cucumber. The cucumber also has what to call the citrate, which is important in healing what to call the acidosis in the kidney. Then we also have what to call the watermelons. The watermelons. Very many others, those fruits that are having a lot of water. The tomatoes can also work. Tomatoes. The oranges, these ones are also very important. So this is the way you can control that problem called acidosis. What is the importance of what to call oxaloacetate? This oxaloacetate here, it is very important in what to call mitochondrial biogenesis, also called the neurogenesis, and also what to call the mitochondrial biogenesis. This means the manufacture of what to call the mitochondria. In other words, increasing the levels of what to call the what? Mitochondria, and also the nervous system, the neurons. It means you must be having what to call these compounds here. And most of them are from what to call the fruits we eat. Don't eat a lot, eat moderately. Thank you very much. I hope with this you can be able to diagnose the problems that affect the members of the community. Thank you very much, Mr. Gos. God bless you.